Hello everyone, today I'd like to talk about recombinant DNA and transformed bacteria, part 2. This video will be a little bit more application based than the last one, so stay tuned. So if we look at this page, on the left we have our sensitive E. coli with no plasmids and a ligase. If we look on the right, there are 4 possible scenarios. We have our regular sensitive E. coli, we also have our transformed with non-recombinant plasmids, and then we have our transformed with only foreign DNA, and transformed with recombinant plasmids. So essentially, the one top here, it's just the default normal E. coli. Down here, it contains an E. coli with a plasmid. And then down here, it's going to be our E. coli with the chromosomal DNA with foreign DNA. Foreign DNA is something that we usually add into the plasmids, as I mentioned in the previous video. And then the last scenario is going to be transformed with recombinant plasmids. This means that the recombinant plasmid has the foreign DNA inserted into the plasmid, and we insert that into the E. coli. As you guys can see on this page, there are 2 main sections, but however, we will only be focusing on the left side for now. As you guys can see, there is a plasmid, which is recombinant, as the foreign DNA is inserted into it. We have OXA and the neo, and they are both selectable marker genes. So in simple terms, basically OXA, which stands for oxacillin, interferes with the cell wall synthesis, while neomycin messes with protein synthesis. So now let's take a look on the right and see the 4 different situations. At the top, you guys see that the positive green refers to the surviving and the negative red refers to dying. You could also think about it as growth versus no growth. So in the first situation, we have our non-recombinant, no plasmid, no foreign DNA, just the regular E. coli with only chromosomal DNA. And in this situation, when placed in either neomycin or oxacillin, neither one of them will survive. This is because the neomycin and the oxacillin will perform their regular functions and cause the E. coli to basically not grow. And in this situation, we have our E. coli with the chromosomal DNA along with the recombinant plasmids. And in this case, both will grow under neomycin and oxacillin. This is because the recombinant plasmid contains the selectable marker genes for oxacillin and neomycin, which means that it will confer resistance to the antibiotics, allowing them to grow. In the third example, we only have our foreign DNA, and in this case, the foreign DNA will only be the oxacillin, the OXA gene basically, and in this case, they will both die. This is because when foreign DNA is inserted into a plasmid, it perceives that as basically a foreign object, and it will try to break it down, essentially bringing it back to the regular E. coli. And then in the last situation, we have our plasmid with recombinant, or we have our E. coli with the recombinant plasmid, but it only has the oxacillin gene, OXA gene, and in this case, it'll only grow on the oxacillin, and this is because the neomycin gene is not there, which means that it won't confer resistance to neomycin, which means that it won't grow on neomycin, but since it has the OXA gene, it will eventually grow on oxacillin. And for a final challenge question, I guess, let's say we have an E. coli with the chromosomal DNA, and then let's say we have a plasmid with non-recombinant plasmids. In this case, what will be the situation for both neo and oxa? Will it either die for both, survive for both, or will it be minus plus or plus minus? Comment down below and see you guys next time.